Hi, this is Shane, and welcome back from the break. It is my pleasure to introduce our next speaker, who is Inez Lam. And Inez is actually a PhD candidate in the Institute for Computational Medicine at Johns Hopkins University. And she is also a Johns Hopkins AstraZeneca scholar. And we had originally invited Inez to speak in the session on diverse contexts for multiscale modeling because of the unique interaction she's had with industry. So thank you for joining. Again, if you have questions for the speakers, just go to the agenda on the wiki. And in this case, click on the heading instead of the title. So in this case for Inez, if you have questions, click on Future of Diverse Contexts for Multiscale Modeling. That will take you to Inez's bio and her abstract. And if you keep scrolling down, you'll see the location where you can enter your comment for Inez, which Grace will read at the end of her presentation. So with that, I'll hand it over to you, Inez.
Hi, can everyone hear me?
Go ahead.
Oh, okay, great. Okay, awesome. So, okay, hi. Hi, everybody. So thank you, Shane, for the introduction. I'm Inez. I am a PhD student at Johns Hopkins, and today I wanted to talk to you about some of my experiences thus far working with researchers in both academia and industry to build a systems pharmacology model of antibody-drug conjugates, or ADCs. So just as a bit of background, I first stumbled across multiscale modeling as an undergrad at Hopkins in a class called Systems Pharmacology and Personalized Medicine. And so this class was taught by Dr. McGowan, who is actually my current PI. And I ended up joining his lab at the Institute for Computational Medicine and really just never looked back. So I was especially struck in the beginning by how many diverse applications of multiscale modeling that there were. And that was the first thing that really came to my mind when I was thinking about diverse contexts for multiscale modeling. So within the McGowan Lab, we use multiscale modeling under a multitude of different biological contexts to look at the impact of different therapies on the diseases. So for instance, we look at the role of vascular endothelial growth factor receptor, or VEGFR, of that system in vascular diseases. We also look at how viral and cellular dynamics affect HIV, how tumor and stromal cells affect cancer, and also immune cells for endometriosis. And although we all work on very different diseases, we all use a lot of the same tools and share the same systems pharmacology and mechanism-based approaches, which means that something that's done in one project within the lab can easily spark ideas for another project. So for today, I wanted to focus on something that's actually unique to my project, which is the context for conducting research. So specifically conducting research in academia and as well as industry. I'm part of the first cohort of students in the Johns Hopkins AstraZeneca Scholars Program, which is a new kind of PhD training program that's aimed at developing researchers at the intersection of both academia and the biotech industry. This happens by immersing the students in both environments. So not only do I work with my advisor and lab at Johns Hopkins, but I also have access to mentors and resources from AstraZeneca as well. And so because these two organizations are really close by each other, I'm lucky to have access to both campuses and am able to develop long-term partnerships with the people from both organizations over the course of my thesis project. So as a student, this setup really allows me to take advantage of the strengths of each of these different research contexts. So on the academic side, I can work on longer term projects, and I have the freedom to explore more big picture or blue sky questions, as well as having access to experts in both multiscale modeling and the disease area. On the other hand, on the industry side, we have obviously lots and lots of data that's available for modeling, as well as expertise from the people who actually develop and make decisions on the drugs. So all the way from the discovery phase to the clinical phases. And these people also have the pressing questions related to the drug, which gives the model a very real world application. And this takes it a step further than perhaps a traditional collaboration, by allowing for more direct feedback and input from both sides in the development of the model. And so by answering the questions that are relevant to both academia and industry, we can have a more direct impact with our multiscale modeling. And of course, there's lots of interest for quantitative systems pharmacology, or QSP, within industry, since it can allow us to test hypotheses and also generate new insights in an efficient and cost-effective manner. And so this is especially important, as we know, for drug development. So as I was designing this project, I was able to meet with leaders from each of the therapeutic areas within AstraZeneca to identify key areas where multiscale modeling could come into play. And that's how I decided to focus on ADCs for my project. So ADCs, or antibody-drug conjugates, are a class of cancer therapy consisting of an antibody, which targets the cancer cells, a linker, which joins the antibody to the drug, as well as an antibody to a small molecule drug, which is actually what kills the cancer cells. And this is also known as the warhead. So the high specificity of the antibody, along with the high cytotoxicity of the warhead, in theory, should allow for selective killing of the tumor cells while sparing healthy tissue. And although the concept of ADCs has been around for many decades, only 5 ADCs are currently on the market in the US. So there is a real need to better understand ADCs and as well as how they function. And ADCs are an especially good candidate for multiscale modeling because they are very, very difficult to optimize. In fact, we need to optimize 3 different components, the antibody, linker, and the drug, the warhead, to determine the overall properties of the ADC. And each of these 3 components allow for different design levers that can be controlled. For instance, the drug to antibody ratio, which is the average number of drug molecules attached to each antibody. We also need to understand the pharmacokinetics and pharmacodynamics of each component, both separately and altogether within the system, within the context of systems pharmacology, as well as understand the balance between safety and toxicity so that we can better predict the therapeutic index. And these models can be applied at any stage of the drug development process, starting from the discovery phase, where we're actually still designing the drug, all the way through preclinical and clinical development. And although application of QSP modeling to ADCs is still relatively new, there's already a lot of great work that's being done by various groups, and I wanted to highlight some of those here today. So first, we have groups at the University of Michigan, led by Dr. Greg Thurber and Dr. Jennifer Linderman, that are working on models of TDM1, which is the first approved ADC that made by Genentech. Another group at SUNY Buffalo, led by Dr. Deval Shah, has created PK/PD models of multiple types of ADCs. And finally, and there are also industry modeling efforts as well from companies such as Novartis and Genentech. And all of these models explore a variety of aspects of ADCs, from looking at co-administration of the ADC with its antibody counterpart, also to looking at different dosing regimens, as well as ADC design scenarios. So this leads us to our systems pharmacology model, which where our goal is to build a multi-scale model of AstraZeneca's ADCs that are attached to a particular class of topic. And ultimately, we want to use this model to better predict therapeutic index and also to better understand the properties of ADCs, their kinetics and mechanisms, any off-target and bystander effects, as well as the factors that have the biggest impact on safety and efficacy. And this can serve as a modeling framework moving forward that can be adapted to other types of ADCs as well. So if we take a look at the overall structure of the model, it's split into 3 scales, each of which are based on the relevant experimental data generated by AstraZeneca. And so we have the in vitro, in vivo, and clinical scales, and we have the computational models that will connect these pieces of data. And we can use these models to then run simulations and make predictions. So at the in vitro scale, we model the cellular and intracellular mechanisms of antibody-drug conjugates and run simulations to see how modifying ADC design characteristics can affect their performance. On the in vivo scale, we can look at tumor properties and systemic distribution to predict bystander effects and also tumor growth and inhibition. And finally, on the clinical model, we can model virtual patients with specific attributes and use those to run virtual clinical trials and test different treatment scenarios as well. And altogether, these will give us a better understanding of the therapeutic index of ADCs. And for today, I'll focus on this in vitro model that I've been working on thus far. And here is a schematic here of the in vitro model. We took a first principles approach to building it based on the known biological mechanisms of ADCs, which includes binding and unbinding of the antibody of the ADC to its antigen, also as well as internalization and recycling, trafficking and release of the warhead into the cytosol, and finally the formation of the effector complex, which is the warhead bound to DNA that induces cell death. And with this model, we can use this to explore the key AD design properties. So, and one example that I'll show today is the drug to antibody ratio that I mentioned earlier, or DAR. So, for this analysis, we wanted to look at the effect of varying the number of the drug molecules per antibody between 1 and 10, which is kind of the typical range that's done experimentally. And we found that as the DAR increases, the amount of the effector complex that's formed also increases, as does the amount of warhead that's available outside the cell that would be important for bystander effects. On the other hand, the cell population decreases exponentially, which means that we get the biggest gain in cell killing from the first few warhead molecules, as shown in the red box. So this suggests that the optimal DAR for this system might be somewhere between 2 and 4 molecules, so that we get the largest amount of cell killing possible without having too much excess drug to cause any cytotoxic or toxic side effects. So that analysis was one example of a key ADC design property that we looked at. We can also use the in vitro model to explore other ADC properties as well, such as the linker design, warhead potency, or also to conduct a sensitivity analysis on the system. And while I won't be covering these for the sake of time, you can find more details about this on my poster. So after building the in vitro model, I can now incorporate the pharmacokinetics to expand this to an in vivo model. And in order to scale it up, we can use the same reactions that are recurring in both the blood and as well as another compartment representing the rest of the body, along with intravenous dosing and systemic clearance. And this can be used to represent blood tumors in which the cancer cells are within the blood. And in order to model solid tumors, we can add a separate tumor compartment that will incorporate the spatial distribution of the ADC within the tumor as well. So to summarize, this in vitro model that we've built thus far allows us to explore the effects of ADC design properties on the system. And this will also serve as the basis for the in vivo and clinical models that I'm working on moving forward. And finally, to conclude, I really believe having these diverse research contexts has really enriched my graduate school experience. So compared to like a traditional internship, because of the long-term nature of this program, I'm able to build long-term working partnerships and relationships and with researchers on both sides, and as well as take advantage of the close proximity between these two institutions. And so I'm really able to take full advantage of the variety of resources and expertise that are available to me. And this program also gives me the flexibility of research exploration in academia, while also maintaining the relevance and the real-world impacts of that industry researchers have. And although having more, I believe that having more researchers that understand both industry and academic research environments will help us build stronger collaborations later on that will hopefully lead to more important discoveries and making the most of both the resources of academia and industry. And of course, I'd be happy to take any questions, especially for any trainees and PIs that might be interested in this type of program or collaboration. And last but not least, I wanted to say a very big thank you to everybody at Hopkins as well as AstraZeneca for their support, and especially a big thank you to my mentors, Phelan and Rosalind. And of course, thank you everyone here for listening, and I hope you have a very lovely St. Patrick's Day. I never knew that this lab photo would come in so handy, but here it is. So thank you very much.